Okay, so it looks like it's just you and me now. We finished off the last lecture looking at the extrinsic and intrinsic pathways of apoptosis, and apoptosis is central to many aspects of uh, our physiology. In the fetal stage, we require apoptosis for our development in the mother's womb. Our hands, for example, are first of all made as a single pad. The fingers then do not grow and protrude out from that pad. Rather, apoptosis removes the spaces in between the digits of what will be our fingers. In many, many ways, apoptosis is a central part of embryogenesis. Apoptosis is essential in wound repair, wound healing. Many repair processes within the body require building and destruction, and apoptosis is one mechanism which is very important in these processes. And as we're seeing now, apoptosis is also essential in immunology. So we looked at two processes, intrinsic and extrinsic, and we saw that apoptosis can occur in a mitochondrial dependent, with release of cytochrome C and formation of the caspase 9 apoptosome, or can occur in a mitochondrial independent manner. And natural killer cells can trigger both pathways. The extrinsic pathway by forming these death receptors, which give a signal to the natural killer cell that this cell needs to be triggered for apoptosis, and also enzymes which are able to penetrate the target cell and then act upon BID. So the next slide will look at how BID interacts with the mitochondria in the mitochondrial dependent pathway. Here is a little map, if you like, of the domain structure of this important target protein called BID. Has a number of alpha helices, numbered alpha 1 through to alpha 8. It targets the mitochondrial membrane via this membrane inserting region. Its name begins from these BH3 domains, which we're not going to talk about today. But we are going to talk about this big area in between here with no alpha helix, which is the target for many different regulatory enzymes. So there are phosphorylation targeting sites, but all these cleavage sites, and in relation to the role of the natural killer cells that we're looking at today, there is a target for granzyme B on aspartic acid 75. In addition, we have caspase 8, which, if you recall, caspase 8 is a part of the death receptor, and caspase 8 also is able to cleave BID. So there are multiple cleavage sites for different proteases. Proteases cut proteins, and they recognize a specific signal site, and they cleave at a specific site as well. So there's more than one of them. Now, why do we need this cleavage? It's because this big membrane inserting region is actually hidden. These alpha 6, 7, and 8 domains are actually buried deep inside the protein, so it's not able to interact with the membrane. Proteins generally are hydrophobic in their core, and this enables them to pack together. And the outer surface normally is hydrophilic to match with their location in the cytosol for a cytosolic protein like BID. Cleavage is required, and step 2 here indicates now that in this example caspase 8, but it's the same as true for the granzyme cleavage. Cleavage here at this big exposed loop in between the alpha helices changes the structure such that these alpha 6, alpha 7, alpha 8 helices, which normally are hidden inside the center of the protein now become very exposed, and these now are energetically much more kinetically. It's much more favorable for them to now interact with the lipid membrane of the mitochondria, and then the rest of the molecule is then removed. So alpha 1, alpha 2 is lost. Okay, so how does BID begin the work of doing something to the mitochondria? First, it's translocation to the mitochondria following this enzymatic cleavage, either by uh, granzymes or the caspase 8 associated with the death receptor. Right, okay. What we're looking at here is the role of a particular virus called vaccinia virus in targeting this process. So it's a different slide from a different paper. It's indicating the granzyme pathway and extrinsic apoptosis by this death receptor. So it's this part is the same as what you saw before. The mitochondria is here. It's the same as what we saw before. Here is BID, which we're focusing in on now. Release of cytochrome. Here we are. Release of cytochrome C, formation of the apoptosome, and activation by cleavage of an effector caspase 9. Now, this figure gives a little bit more detail about some of the cellular components here. But what want you to focus on is these big red circles here. Apoptosis is so effective in dealing with viral infections that in order for viruses to be able to survive and to continue their spread in their target population, they must have mechanisms of overcoming apoptosis. And I think all viruses, as far as I know, have some genes which have the capacity to interfere with apoptosis and prevent or slow down apoptosis. So this is just one example of one virus, vaccinia virus. If you recall the monkeypox virus outbreak, which reportedly began in 2022 and then spread all over the world, it caused very few deaths. I think it did cause some deaths, but it fortunately was fairly it died out. One of the reasons it was easily controlled was because vaccinia virus is actually an effective vaccine against monkeypox virus. We're all members of the same family. Vaccinia is a very well studied virus, and it blocks a huge number of aspects of this pathway. It's incredible. It has a gene for a protein called F1, and this blocks these BCL3 member proteins here, which are essential for the function of BID. It also has a protein called N1, which directly it affects these proteins, which are all members of the same family here, but it also directly inhibits T-BID. T-BID is the truncated form of BID, this one here. So it basically prevents T-BID from being able to translocate to the outer mitochondrial membrane. It has also F1, which is up here. F1 has another function, and it blocks the activation of procaspase 9. The caspases are produced in a pro form, all of them, which are unable to, which do not have proteolytic activity, but they are processed by cleavage by another caspase further higher up the chain. It's like the thrombin blood clotting cascade, similar kind of concept. So F1 prevents procaspase 9 from being cleaved and matured into the apoptosome. F1 also is able to block the activity of the active caspase 9. And we have many other proteins which are blocking all different aspects. Also, this is interesting. There's something here from vaccinia which is able to form a channel in the endoplasmic, or in the Golgi, sorry. And I said the endoplasmic reticulum because the endoplasmic reticulum is the major source of calcium in the body. And so an endoplasmic reticulum or Golgi, this channel causes a calcium which is normally present at a very high concentration intracellularly here. It causes this calcium to constantly leak out, and that prevents calcium from acting as an important second messenger in the apoptosis pathway, which depends upon a sudden elevation of intracellular calcium. And this is just one example of one protein, one virus, which is able to interact with all of these different aspects of the pathway. If we looked at coronavirus, we'd also see a number of ways in which coronaviruses block apoptosis, and we could do that for any virus. Okay, so although this section of the lectures is a general overview focused on the role of cells, there are some aspects which are very important, but which will not be covered in detail in the other sections. Which, for that reason, I've done a deep dive into them, and this is one of them, apoptosis. We'll not talk much, if at all, about apoptosis again, and so I've introduced it here in relation to natural killer cells. So now we're going to recap. This was doing a deep dive. We're going to come back to the surface now, and look at this in a more overview type of a way again. So we were looking at this, basically, the role of the natural killer cell in causing apoptosis in target cells through triggering the extrinsic pathway via the death receptors, as well as releasing granzymes, injecting granzymes directly into the cell in order to trigger the intrinsic pathway. And you might ask the question, if you think about it, there is a question here. And that question is, if the cell is infected and it needs to undergo apoptosis, and if there is an intrinsic pathway, which we said there is, which goes via mitochondrial release of cytochrome C, then why do we need natural killer cells? Why do we need that? If this apoptotic cell knows it's apoptotic now in this picture, but if the infected cell knows it's infected and is able to form the death receptor on its surface to bring the natural killer cell, rather than that, why doesn't it just trigger its own apoptosis? And cells are capable, any cell is capable of triggering its own apoptosis under the right conditions. And the answer is that, first of all, in our natural physiology, apoptosis generally requires signals from outside. If, for example, in the development of the digits on our hand, our fingers, if this requires apoptosis, so how does a cell know that it should undergo apoptosis? So some of the cells in the digital plate of our hand, which is developing during embryogenesis, must undergo apoptosis, whereas other cells will become our fingers, which should not undergo apoptosis. They cannot know unless they have positional signals which come from outside of the cell. So an interplay of signals from outside of the cell are necessary to direct the required cells for apoptosis. So from its very basis, the cell is set up in order to receive signals to direct it to undergo apoptosis. Now, coming to the specific instance in immunology where the cell needs to respond to infection, the concept is that multiple signals are required to be certain that uh, apoptosis is what is necessary. And this is even a common thing in life. If I mark someone, when I mark exams for students, for example, I go through the marking process and then I have a rest and I check again the marking. And I often give students a chance to interact with the material. I send them the list of correct answers and give them a chance to respond even. And it may turn out that I made a mistake somewhere and I can correct it. Uh, we don't want to make mistakes and we don't want to kill cells that are actually not infected. How dangerous the immune system is obvious from uh, autoimmune diseases and uh, extreme allergic reactions. There are many conditions where the immune system becomes our own enemy. Uh, it's carefully balanced, and uh, this balance between being effective at killing cells and also um, not killing ourselves means that there's some middle ground where some diseases must naturally occur. Uh, but if we erred too much on one side or the other, we would be in a situation where we're unable to respond to infection, and we'd all die in our infancy, or we would die or be severely harmed, all of us, by having so many uh, constant allergic reactions. In order to maintain this balance, uh, effective functions of immunology require multiple input signals. So the cell knows it's infected, but to be sure, it waits for the natural killer cell to come and give this signal. And the natural killer cell responds to multiple targets. So it's not just having the ligand for the death receptor ready to see the death receptor. If that was the case, then the question would be valid. Okay, so what's the point of the natural killer cell? The natural killer cell has the death receptor ligand, but it's not in an active, the cell itself is not very active, it's very sleepy. And it requires these sentinel cells called macrophages to release signals to activate it. In other words, this natural killer cell is being told in at, in more than one way um, that there is an infection. So it can see this death receptor, but it doesn't care. However, if macrophages also are releasing factors such as the interferons, which we haven't spoken about before, but these are very important molecules that recognize intracellular infections. So primarily we're talking about viruses, although there are some intracellular bacteria as well, which can trigger this. So the macrophage releases interferon alpha and interferon beta. So now the natural killer cell, if it's getting a signal from these macrophages via the interferons, and it's also seeing death receptor, then this is a confirmation that uh, this tissue is in an infected state, and the apoptosis of any cell with expressing death receptors is actually truly required. Uh, and there's also a third factor, and this we'll talk about in the section on innate immunology, but we'll mention it now. And this is that in innate immunity, there are some general signatures which are indicative of an infection. The most well-studied example is lipopolysaccharides in gram-negative bacterial cell walls, which are present obviously during infection with a gram-negative bacteria like E. coli or Salmonella, amongst many others. Uh, those are called pathogen-associated molecular patterns. Those signatures are called pathogen-associated molecular patterns, and those are recepted by uh, pathogen recognition receptors, or to be precise, PAMP, pathogen-associated molecular pattern recognition receptors. On the surface also of natural killer cells. So natural killer cells are receiving two innate signals. Macrophages are releasing interferons. Pieces of the pathogen themselves may be in the environment of the natural killer cell and may also serve to greatly amplify the the response of a natural killer cell. And we're talking about responses 100-fold more. So you can take these cells in vitro and watch their behavior, and you can measurably see 100-fold increase in activity of that cell once it's receiving these innate type of signals. So all of this is happening in destruction of target cells without any of these immune cells or the target cell actually knowing anything about this specific invader. That comes when we talk about the adaptive immune system, T cells and B cells. So here we have this axis now, which leads to apoptosis of target cells. In addition, natural killer cells are also able to release an interferon called interferon gamma, and it's like a cycle here. Interferon gamma actually also activates macrophages, so they can activate each other. So we have this amplifying signal here. Macrophages can activate natural killer cells, and natural killer cells also activating macrophages. And the activated macrophage is now also releasing various cytokines. So this one is important in T cell polarization. T cell polarization means making it actually capable of being uh, of being cytotoxic or of actually going and performing its helper functions or regulatory functions. In addition, it increases the phagocytic potential of the macrophages. So the macrophage is a little bit sleepy, but if it receives signals, interferon gamma signals, then it wakes up and becomes much more alert and much more active in engulfing microbes. Engulfing microbes. Now we come to the other sentinel cell, the dendritic cell. So these are the three sentinel cells that we looked at: the macrophage. Actually, I said the three. I mean, because the mast cell is the third one. It's not on this slide. The dendritic cell. But these two are the antigen presenting cells as well. And this is the most professional antigen presenting cell, the major one, the dendritic cell. It also responds to interferon gamma, and that greatly increases its ability to present antigens to T cells. Okay, so when we want to understand immunology, the connection between everything is as important as understanding what's happening with individual cells. And that's what makes immunology such a exciting field, but also quite a difficult one to learn. Takes a while to conceptualize the whole process, and still, our understanding of immunology is subject to continued development and growth. Yeah. Okay. All right. So still on the natural killer cell. We've said that the natural killer cell is an innate cell. It's not recognizing anything specific in terms of a specific pathogen. But it has some aspect to it, which is in a sense adaptive. And this aspect is memory. Memory. We have for B cells which make antibodies, once the B cell has been activated by antigen, it forms plasma cells which make antibodies, and it forms memory B cells which, after clonal expansion, remain ready to fight infection again. And these two aspects, clonal expansion and memory, we actually find them in natural killer cells as well. So that aspect is something adaptive. The way the difference is that Okay, the first thing I need to tell you about the natural killer cell is that it has receptors on its surface which are specific to a class of things, not like the T cell receptor or B cell receptor, which is specific to even a specific subtype, or may recognize, for example, if we take the example of coronavirus, which most people are familiar with, can distinguish between uh, different strains uh, of Omicron. Or now we have the descendants of Omicron. Our adaptive immunity in terms of antibody recognition distinguishes between different lineages of the same virus to such an extent that a vaccine that may be effective at one point after several months may become much less effective because the virus has evolved a few mutations, key mutations, which uh, enable it to escape immune recognition by those antibodies. Natural killer cells have receptors that recognize um, antigens. However, its recognition is much more general than that. Whenever a natural killer cell has been effective in recognizing something and has been stimulated as well, then those ones which have that receptor undergo clonal expansion. So here we have an example of one receptor called Ly49H, which can be expressed, or minus means the cell isn't expressing that one. And if those get stimulated, then they undergo clonal expansion to become effector natural killer cells. And then not all of these cells actually will survive, but some of them will be laid down as memory cells. So once natural killer cell responses have been initiated in the future, then we can have this secondary response where we can much more quickly have a strong reaction to that. So that's an adaptive aspect of natural killer cells. So mostly we think of them in the way which I described just in the previous slides as innate cells, but they have these aspects of um, adaptive adaptive immunity. And there's a nice review came out a couple of years ago. This is an excellent journal, by the way, if you want to go into any immunological subjects in detail. Annual reviews of immunology, it's yearly, and they'll summarize some of the major developments in different aspects of immunology that have occurred in that year. So that's a good, very good journal to read from time to time. I will remind you of something I said earlier in a previous lecture, that natural killer cells, the name is unfortunate. Initially, we appreciated these cells as cells which are able to have a killing function. Especially, we look now at the example of virally infected cells, but also cancer cells. So this is a natural killer mechanism. It's looking at general signals in relation to misexpression of MHC molecules, uh, in addition, of course, to the cells which happen to be expressing these death receptors as well. So various ways the natural killer cell can see that something is wrong and go and target that cell for destruction. However, natural killer cells are also inhibitory, and this inhibitory function is important in vital uh, physiological processes, such as going back to the beginning of embryology, preventing an immune reaction from occurring against the blastocyst and the developing fetus later on. We know that there is an immune barrier through the placenta. However, that immune barrier by itself is not enough. The immune system is downregulated in order uh, to make sure that no immune cells manage to slip past and start attacking the foreign body sitting inside its embedded into the mother's uterus. And natural killer cells play an essential part in that. They recognize some what we call non-classical MHC molecules expressed on the surface of the implanted embryo, uh, which tell the natural killer cell to release inhibitory signals that prevent other immune cells from causing harm. So that's the first um, of the three lymphocytes. What are the other two lymphocytes? Well, those are the T cells and the B cells. I have very little to say about these now because we'll have a whole section devoted to both of these cells. So we'll only just take a very surface look at them. Uh, these cells are considered to be firmly in the adaptive immune system. Again, if we have a T cell receptor, we also have co-receptors. Co-receptors are not shown for B cells, but actually B cells also have co-receptors. We have lots of them. Okay, give you some numbers here, but those are very, very big numbers. And the amazing thing about these cells is that these receptors, the B cell receptor, it's called the B cell receptor when it's on the surface of the B cell. In its released form, then we call it an antibody. So antibodies are the same as the B cell receptor, except that the B cell receptor has a little tail, which enables it to be embedded into the membrane. Once a B cell recognizes an antigen, then that B cell becomes activated and can form. Okay, it becomes activated. There's clonal expansion, and it can form either plasma cells that release tons of antibody or memory cells that will maintain the ability to respond quickly and powerfully in a future infection. And there is a massive and amazing diversity in these receptors. So we have, for example, 3 billion B cells circulating in the blood. The theoretical diversity of B cell receptors actually is more than this number even, which means that we have this huge mixture of B cells all expressing different variations on the B cell receptor, which are able to recognize potentially any pathogen that may come to infect us in the future. And obviously, we'll talk a lot more about that and how that happens when we come to that section. Antibody production, neutralization, opsonization, two terms in immunology. Neutralization means that the antibody produced is actually able to block the infection. Opsonization means that we coat the surface of the pathogen, but it's not destroyed directly, and it's not prevented from entering cells in the case of a virus. But other factors come and then destroy that pathogen. So, for example, if you take in a test tube antibody, virus, coronavirus, if you like, and host cells, and mix these three together, then if you have a neutralizing antibody, there will be no infection. But if you have an opsonizing antibody, there will be infection. However, in the body, we have many other systems, macrophages and the complement system, as well as other systems, which will recognize these antibodies bound onto the target cell and will get rid of it. But if we have in isolation just the antibody, the host cell, and the pathogen, say the virus, those three things in the test tube, then neutralizing antibodies will effectively as if they're killing the pathogen. They don't actually kill it, but they prevent it from being able to replicate in or upon host cells. For the T cell receptor, it always stays firmly bound onto the membrane. There's no soluble form. Like the B cell receptor, we have a huge diversity in the T cell receptor, and it's for killing virus-infected cells. So that would be a cytotoxic T cell. We also have another type of a T cell called a helper T cell, which actually is required to assist B cells in mounting a response. And we also have a third type of B cell called a regulatory T cell. We usually call these Tregs from reg, CD4 positive T cells or cytotoxic T lymphocytes. And these ones we call CD4 positive cells or T helper cells. I'm not going to ask you this question. Okay, so finish the review of the cells now. Finally, in this final section of slides in this first section of overview, we'll look at the anatomy of the immune response, where things are happening. We looked at it briefly when I indicated to you the lymphatic tissue, the hundreds of lymph nodes that we have, 200 or 300 lymph nodes in our body, and the difference between primary and secondary lymphatic tissue, the bone marrow and the thymus. We will look at this in a little bit more detail now. Okay, so what we have here is a kind of a stylized representation of the cardiovascular system, blood flowing through our body. And we have the lymphatic system. So in the lymphatic system, we have lymph nodes. And this is where T and B cells actually learn from dendritic cells and also macrophages and other cells present in the lymph node. This is where they meet antigen for the first time. How did they meet antigen for the first time? Because in inflamed tissue, then the lymphatic system there drains from there to the nearest lymph node. And at the lymph node, that's where the T and the B cells are sitting, waiting to try out their T and B cell receptors to see if they're able to recognize anything that are being brought from incoming dendritic cells for the most part. So the dendritic cells are waiting here, sampling all possible infections, and then carrying the pieces of microbe to the lymph node. If a recognition happens, then that T or that B cell becomes activated. The T and B cells, those are circulating in the blood, and they reach the lymph node through what are called high endothelial venules. So it's a anatomically slightly distinct type of a vein, which is very specifically capable of allowing the extravasation of T and B cells. So T and B cells reach the lymph node from the blood, and the professional antigen presenting cells, dendritic cells, reach it via the lymphatics. Of course, the dendritic cells initially were in the blood, and they reach inflamed tissue, and many of them then tissue resident. So initially, of course, all of the leukocytes, so on myeloid or lymphoid, circulate in the blood, but some of them enter the tissues and are waiting for infection, and then they travel via the lymphatic to the lymph node, whereas the lymphocytes travel directly to the lymph node and enter the lymph node via these high endothelial venules. We use the word in physiology afferent and efferent. Afferent is coming in, efferent is coming out. So we use it in the lymphatic system and also in the cardiovascular system, afferent and efferent. So afferent lymph node, efferent lymph node. Lymphocytes actually travel through the lymphatic system as well. Once they've entered the lymphatic system, they circulate throughout the lymphatic system. If it sits here in the lymph node and doesn't see any antigen for a long time, it gets bored and says, well, there's no fun here. There's nothing happening. Let's go and see if there's anything interesting in another lymph node. So there's a circulation also of lymphocytes through the lymphatic system. But eventually, what's in the lymphatic system drains back into the blood system via the thoracic duct, which is situated above the heart, um, in order for another cycle to go. So this is the connection between the two systems. The lymphatic system rejoins the circulatory system via the thoracic duct, and throughout our tissues, plasma, fluid, uh, some solutes, as well as immune cells, leave the circulatory system uh, to enter tissues throughout our body. And now there's one thing that's new in our discussion, and that's the spleen. The spleen is an important tissue, and its role is actually to screen for bloodborne pathogens. Bloodborne pathogens. So uh, for this course, most of the time I'll be focused on what's happening in lymph nodes, but also there's a screening process for the blood. There are many bloodborne pathogens, and uh, lymphocytes also sit in the spleen and are concentrated here, and antigen presentation is also happening here in the spleen. And this is where T and B cells can become activated in order to deal with any bloodborne pathogens. Some pathogens, once they get into our body, they don't sit in the tissues. They travel throughout our blood. So this is a place where we have important screening as well. I mentioned about lymphocytes recirculating all the time, even in the absence of infection. An interesting number here, huge number. Every minute, 5 times 10 to the 6, 5 million lymphocytes leave the blood and enter secondary lymphoid tissue every minute. So much activity going on. Even if we're just sitting down quietly, attending an immunology lecture, our bodies are very, very busy all the time. And here is another overview slide. And this one also introduces the idea of the primary and secondary. So we've seen that before in the picture of the human body, where things are happening. Here is more indicating conceptually what's happening. It's a summary slide. There may not be much new things here. Thymus and bone marrow. SC, this stands for stem cell. So these are those hematopoietic stem cells that we saw in the first lecture. The lymphoid, common lymphoid progenitor can produce B cells and T cells. Both B cells and T cells begin their life in the bone marrow, but indicated by the fact that the stem cell is in the bone marrow. But the T cell then migrates to the thymus, and it completes its differentiation there and there. Education means becoming immunocompetent, able to recognize antigen. So that immunocompetence means expressing a T cell receptor, or in the case of B cells, expressing a B cell receptor. We will look at how B cell receptors get made and expressed. It's not as simple as you might think. It's quite a story for the B cell to express this receptor, because it has this unique feature, as I mentioned, that there are millions or billions of potential of different variants of B cell receptor, and not all of them are functional. So there's a genetic mechanism that creates this huge diversity, and these receptors have to be tested first to make sure that they're actually functional and able to even associate together. Antibodies made up of 2 types of chains, heavy chain and light chain, and they have to, and 2 heavy chains have to come together, and each heavy chain is associated with a light chain. These have to be tested to make sure that they're actually effective. Then, in terms of immunocompetence, before we let these cells loose to go and potentially cause destruction in our own body, there's a mechanism for testing them to make sure that they are not self-reactive. And we call this subject central tolerance. And we'll look as well at the mechanism of central tolerance when we come to it. So this is the education stage. At the end of this stage, we have an immune competent T or B cell. It's not yet seen any foreign antigens, but it has a T and a B cell which is all ready to go, and it's safe. So it has a weapon. The safety is on, so it's not going to shoot its friends, but it's armed and just needs to see the enemy in order to perform its effector functions. Where does it see the enemy? In the secondary lymphoid tissues. So far, I mentioned the lymph node and the spleen. The lymph node recognizes antigen in tissues, and the spleen recognizes antigens in the blood. These are what are called encapsulated lymphatic tissue, in distinction from what are called unencapsulated. Here we have these tissues are surrounded by a capsule. Most of our organs have a tough shell around them, a capsule. Regard whether they're lymphoid or other. Kidney, for example, if you ever tried doing a dissection and you want to get into the tissue, there's a very tough, fibrous, cartilaginous film, a thin layer, very, very thin, but yet very tough and very resistant. And this encapsulates the whole node or the whole spleen. We have other tissue, and this tissue is at mucosal surfaces, and we call this molt. And I will talk about that in details as well in coming lectures. This is not encapsulated. Basically, it's a collection of immune cells which are interdigitating. Maybe I can find an easier word than that. Which are mixed in with epithelial cells and fibroblasts from the organ. So, in the terms of, for example, the gut, there's something called the Peyer's patch. So in our digestive system, there's a collection of immune cells which are mixed in with the normal cells that are functioning to help in the digestion of our food, the release of various enzymes which digest food, release of mucus that makes a lining to the from where the word mucosal surface comes from, as well as having the function to actually import the digested food into our body and let it cross into the bloodstream. So this is the natural digestive system cells, epithelial cells, and mixed in with them we have a dense collection of immune cells. So we can see that there's something special. It's distinct from other parts of the digestive tract. So we have patches, areas where there's no distinct clear edge. With the encapsulated secondary lymphoid organs, there's a clear finish, a clear edge to the thing, a distinction where the kidney begins and where the kidney ends. There's no doubt about it. But here, there's no distinct clear beginning and end. But when we look back, we could, at a distance slightly, we can see that, okay, there's an area where there's a difference in texture and appearance. Here is a slide from a very nice textbook, which I didn't mention in the beginning. This is Abul Abbas. It's actually a great textbook. I took this slide from him, and it's about the thymus, and something that's concerning perhaps as we're getting older, that there are changes to the output of the thymus. As we get older in years, our thymus releases fewer and fewer T cells. And after about the age of 30, we reach a minimum level, and the thymus is releasing T cells out throughout most of the course of our life, but much fewer than when we are younger. Furthermore, the distinction between naive T cells and memory T cells also changes. So when we look at those T cells that are released from the thymus, so here we have a lot, but here we have a few. Now we're looking at percentages. So percentage of this thymic output. Early on in life, most of the T cells which are immunocompetent released are naive T cells. They've never yet seen antigen. As we get older, the level of memory T cells increases as the level of naive T cells decreases. So as we get older, we have a lot of memory to past infections, which is good. In a sense, it's natural, isn't it? Because if we've just been born and we haven't been exposed to antigens, why would we have memory T cells? We wouldn't, would we? Because we haven't seen anything yet to remember. But as time goes by and we accrue more infections, so we get more of these. So that's quite natural. But this drop in relative numbers of naive T cells, along with reduced thymic output, or very significant reduced thymic output as we approach our 30s and onwards. What that means is that older people are less able to mount a T cell response to new infections. So if you're exposed to SARS coronavirus 2 in your 60s or 70s, you're much less capable of mounting a good adaptive immune response than if you're exposed to this in your youth. And of course, there are some viruses which are particularly deadly to young people or to children, but more generally, for new infections, we have a much harder time dealing with them in older life. And that's the true whether we talk about a natural infection or a vaccination. The response is going to be lower. We have going to have fewer naive T cells to potentially recognize this new type of infection. And what that means is that we'll have a poorer T cell response. It also means that we'll have a poorer T helper cell response. So it's even for B cells, we won't be able to mount such a good response because B cells need T helper cells to produce good quality antigens. B cells can produce antigens. I meant to say antibodies. B cells can produce antibodies without T helper cells, but they don't produce a very sustained or effective antibody production without T helper cells. I think that'll be enough for today. Do you have any questions? Okay. Good. I'll post this onto YouTube as well in a while too, so the other students can see it as well, and you can review it again. Good. All right. Look forward to seeing you on Thursday, inshallah. Okay. Take care, everyone.